Thank you very much, Stephen. I'd like to thank the organisers for the kind invitation to tell you about some of the work that we do in my laboratory. Philippe asked if I could speak on the topic of why do we need to understand microbial pathogenesis? And I have to say, as a researcher that's worked on this topic for the last 25 years, that was quite a searching question for me, because there are, of course, some infectious diseases that we have been able to bring under control using relatively simple interventions such as antibiotics and simple vaccines. But this audience, of course, needs no reminding that poultry remain afflicted by diverse pathogens that can constrain productivity and welfare. And of course, they can also transmit through the food chain and farm environment zoonoses that affect people and exert substantial costs to the economy and society. And for some avian pathogens, simple interventions that solve the problem aren't available. And even where we do have control strategies, these can sometimes be limited in their cross-protective efficacy, partly because pathogens evolve rapidly to escape vaccine-mediated protection, and they can also exhibit a high degree of antigenic diversity. Of course, we're also seeing the effectiveness of antimicrobials blunted by the emergence of transmissible drug resistance, and there are restrictions on antimicrobial use in some countries, which will require some more inventive thinking around control strategies for diseases. And I also think it's important that we study microbial pathogenesis, important to train the next generation of researchers to study avian diseases and zoonoses in the animal host where they are a problem. A great deal of published research relies heavily on cell lines and indeed surrogate rodent models, and I believe there's a vital need to be able to study these diseases where they exist. So in my team, formerly at the Institute for Animal Health and now at the Roslin Institute, we study a number of bacterial avian and zoonotic pathogens. Campylobacter and Salmonella you've been hearing a lot about already this afternoon. We also study avian pathogenic E. coli and Shiga toxin-producing E. coli that I won't talk about because they affect ruminants. And as we've been hearing, these organisms collectively account for around 70,000 laboratory-confirmed cases of foodborne illness in the United Kingdom each year. It's probably more like 1% of the national population. And that comes at a high recurring cost to the national economy. And when you scale it in global terms, there's a very substantial burden in terms of human health and healthy life years lost to these pathogens. And as you heard from Lisa and others this afternoon, in certain circumstances, and depending on factors both of the microbe and host, you can also see disease in poultry associated with some variants of these bugs. So I'm going to talk primarily about Salmonella and Campylobacter this afternoon, and I won't reprise all of the background on Salmonella. You've heard a lot about it. Most infections in warm-blooded animals caused by Salmonella enterica subspecies 1, of which there are thousands of antigenically distinct serovars. And those serovars can vary in their host range in clinical presentation, where serovars like typhimurium are largely promiscuous and can therefore cause zoonotic infections. And at the other end of the spectrum, there may be host-specific serovars, which tend to be more associated with systemic disease, such as fowl typhoid. As you've been hearing, handling of contaminated poultry meat and eggs remains an important risk factor. And we've seen the evolution of these organisms punctuated over the years by the emergence of epidemic and drug-resistant variants. And so it's a rather moving target. We heard from Stefan de Vries this afternoon about inactivated vaccines, which are partly credited with reducing the incidence of non-typhoidal salmonellosis in humans. This graph shows the pandemic of Salmonella enteritidis phage type 4 and other prevalent Salmonella serotypes during the 1980s and 90s. These are data from the UK. And the point at which we began to vaccinate broiler breeders and layers and indeed implement other control strategies was somewhat coincident with a major reduction in the incidence of human infections. But I should say that it's been rather difficult to discern precisely what contribution vaccines made to this decline because national control programs in the UK mandated the use of those vaccines at the same time as a raft of other control measures. Stefan presented, I think, some compelling evidence for protective efficacy against serogroup B C, and D strains with their trivalent vaccine, but there are other published studies in the literature which perhaps provide a less compelling picture of their ability to reduce gut colonization and egg contamination. And we know that in some countries, even where those vaccines are used, there continues to be a problem with salmonella in poultry meat and eggs. So I believe there's an ongoing need for us to try to devise improved control strategies because we haven't yet solved the problem. So another important point to make at this juncture is that not all salmonella are the same. I've mentioned antigenically distinct serovars that can cause either enteric disease or systemic disease, but we're also beginning to learn that within serovars of salmonella that we long regarded as promiscuous, there are some pathovariants which appear to be host-adapted. There are certain types that cause systemic disease in pigeons and apparently nothing else, and others that cause systemic disease in passerine birds. We and others have begun to use machine learning to try to look at the blueprints of Salmonella typhimurium strains and associate that with their hosts of origin. And it does appear through machine learning that there are pathovariants of Salmonella typhimurium that differ in their invasive potential and their host tropism. So it remains a challenging organism to try to control because of its diversity. Now, we're greatly aided in the task of trying to control Salmonella by the availability of complete genome sequences. And just before I came away, I checked Enterobase, and there are now 197,000 confirmed genome sequences for Salmonella enterica strains. So we have no shortage of genetic blueprints for these bacteria. What we do have, though, is a relative lack of understanding of the role that those genes play in colonization, pathology, and transmission, in particular in farm animal reservoirs where these bacteria can be found. We understand little of when, where, and at what level they're expressed. And because of these two limitations, we also find it hard to interpret the impact of genetic variation, whether that's in the repertoire sequence or expression of genes. To some extent, we can predict through homology how the encoded proteins act, but for many of the factors we find to be important for infection, we don't understand how they work. And then, as I highlight in red, we have to think, how are we going to use information about the function of these genes for some sort of practical gain? And an important priority for my group has also been to answer these questions with minimal use of animals in experiments and minimum harm. So each Salmonella enterica strain has about 4,500 genes. How do we go about assigning functions to those? Well, we're fortunate that Salmonella is a genetically tractable organism. It's relatively easy to introduce mutations into the Salmonella genome and then to evaluate the phenotypes of those mutated strains relative either to the isogenic parent or a repaired strain or one in which the mutated gene has been reintroduced in trans. And you can do this gene by gene. This was a study from my group over 10 years ago now, shortly after the Salmonella enteritidis genome was first published. You can look at the blueprints and identify on the basis of homology candidate virulence factors. Shown here are the 13 fimbrial operons that determine these hair-like appendages on the bacterial surface. These often mediate interactions with host cells, and so we chose in a systematic way to mutate each of the major fimbrial subunits and to test those mutants one by one. And some of them are attenuated, and we can partially restore them to virulence by putting the genes back. Now, that gene-by-gene analysis is powerful, but with an organism with 4,500 genes, that's going to be an incredibly time, labor, and animal-intensive process to assign functions to the genes. And it was with that in mind that at the Institute for Animal Health some years ago, we began to use a method for screening pools of random mutants of Salmonella in farmed animals. This is a technique we call signature-tagged transposon mutagenesis, or STM. As you'll see shortly, this is now superseded by a more powerful technology, but in the mid-1990s to 2000s, this was the state of the art. Briefly, what we do is randomly mutate the bacterial chromosome with a transposon. It's a kind of jumping gene that inserts almost at random into the bacterial chromosome. And we use a panel of transposons that each have a unique DNA sequence. This is the signature tag, and you can think of it as a kind of barcode. When the transposon with a unique tag inserts into the genome of, in this case, Salmonella, it gives that Salmonella a unique oligonucleotide sequence that we can scan for. There are actually 95 of these, but for simplicity, just 4 are shown here in different colors. And so we can introduce these different tag transposons, create mutants, and then take one mutant with each of the different tags and assemble them into pools. And these pools can then be screened in animals, and we can compare the composition of the inoculum with the composition of mutants that we recover from these animals. We call this the output pool. And what we're looking for here is the presence of those organisms by virtue of the presence of the tags they contain. And we do that by PCR amplification of the tags and detect them by hybridization. And what we hope to find, I'm not sure if you can see the laser pointer, is that there are some mutants that are represented in the inoculum, like this one in red, which are negatively selected and don't survive in the host. And we can infer from that negative selection that the mutant lacked a gene that was important for colonization of the host. And we can then find out where that mutation is located by subcloning and sequencing the transposon flanking regions. Now, we did this for Salmonella typhimurium in cattle, pigs, chickens, and mice some years ago. Data are all published now. Screened a relatively modest library of just over 1,000 mutants, but it identified over 200 new Salmonella virulence factors at the time, including things that appeared to be putatively host-specific. Now, this was a very powerful method, but it has its limitations. It's very subjective, comparing the intensity of the hybridization signals between the input and the output. It's very time and labor intensive to make and screen the libraries. And also, even once we've identified negatively selected mutants, it's a time-intensive process to find out where those mutations were located in the genome. And so in the late part of the first decade of this new millennium, we started a project with the University of Cambridge to apply what is now the absolute state-of-the-art to functional annotation of bacterial genomes. This is called transposon-directed insertion site sequencing, or TRADIS. It's sometimes also called TNSEQ, or transposon sequencing. Now, don't be frightened by the detail of that. It is conceptually very similar to what I've just described. We make random transposon mutants where a transposon jumps into the Salmonella chromosome, and we take pools of mutants and screen them in animals. And again, we compare the content of the pool that went into the animal with the content of the pool that came out. It's just that in the case of TRADIS, instead of looking for signature tag mutants, we use a special kind of sequencing called massively parallel sequencing using the Illumina platform to give us 2 types of information simultaneously. We can tell to the nucleotide where the insertions are located, because as we copy out of the transposon, we sequence the adjacent genomic DNA. And because of the massively parallel way in which the sequencing works, the number of times that we return those specific sequence reads then becomes a measure of the abundance of that mutant in a pool. So in this hypothetical example of an in vivo screen, there are 2 genes, A and B, which were each mutated 3 times. The insertion sites are denoted by these red pins. And when we apply this massive parallel sequencing approach, we can see to the nucleotide where those insertions are located within those genes. So returning sequence reads at these positions tells us where the insertions are located. But because of this massively parallel nature of the sequencing, the number of reads is a measure of the abundance of the mutant. And so in this hypothetical example, gene B was mutated 3 times, and there is an equivalent number of reads before and after screening this library in a hypothetical animal. Now that tells us that this gene is not under selection in vivo. By contrast, there's another mutant here in gene A, in fact, 3 different mutations at these positions. The sequence reads in this instance are returned in the inoculum, but they are largely missing in an output pool from this hypothetical experiment. That tells us these mutants were present in the inoculum, but they're missing after infection, and therefore this must be an important gene for survival in the host. Now, this has proven to be a fantastically powerful approach for assigning functions to Salmonella genes with minimal animal use. The data are published some 5 years ago now, and I won't go into all of the details, but we've made a comprehensive library of Salmonella mutants and screened about 8,500 of those in calves, pigs, and chickens. The Salmonella chromosome is here in yellow. The red lines around that denote the position of all the transposon insertions around the chromosome and the 3 strains plasmids in this strain. And we've screened this library of about 8,500 mutants in calves, pigs, and chickens by the oral route. It's the same library, just screened in 3 separate hosts. And remarkably, this methodology was able to give us both the identity and the phenotype for 90% of all the mutants that we screened. So that's phenotypes for about 7,700 different mutants, and it represents about 2,700 different Salmonella genes, so well over half the Salmonella chromosome that you can mutate. So if you calculate fitness scores, by which I mean the log 2-fold change in the number of sequence reads between the input and the output, and then plot all of those fitness scores from the lowest to the highest, you end up with plots like this, where we have a fitness score plotted here for all 7,700 or so of the different Salmonella mutants for which we were able to get the identity and phenotype. And we see these S-shaped curves in all the species. And what it tells us is that the vast majority of mutations that we can make in the Salmonella typhimurium genome have no effect. The fitness score approximates to 0, and therefore these mutants are neither positively or negatively selected in the host. And this indicates a great degree of functional redundancy in the Salmonella genome. There are many genes and pathways that can be removed without having a negative effect on bacterial colonization. But at the tails of the distribution, you can see some that are strongly negatively selected, and that's interesting information because that tells us which genes have been mutated that lead to a loss of virulence. Now, obviously, you have to have clever statistical approaches to know how to approach numerical data of this kind. I won't go into the data, it's all published, but basically, we can apply statistics to find a cutoff in the fitness score that will describe attenuation with some degree of statistical reliability. There's actually a bimodal normal distribution of the fitness scores. If you look at this population, the vast majority belong to this larger population with a fitness score that approximates to 0, but at lower fitness scores, there's another population that are negatively selected. And where these cross, we can say what an appropriate fitness score is to say that plays a significant role in infection. And when you apply a cutoff like that, you end up with numbers such as these, where there are around 600 different Salmonella typhimurium genes that play a significant role in colonization of the guts of chickens, pigs, and calves. These are obviously fantastic targets for the design of control strategies. That might be fitting inhibitors or developing subunit vaccines or using mutants in those genes as live attenuated vaccines. But interestingly, what we found doing this analysis is there are also smaller subsets of genes that play an apparently host-specific role. There are some that are important in chickens that don't seem to play a role in calves and pigs. And of course, that's vital information if you're trying to devise a control strategy, because something that works for one animal will not necessarily work for the next. Among the host-specific factors that we identified in poultry were lots of genes and pathways related to anaerobic metabolism, and that probably speaks to the distinct ecology of Salmonella typhimurium in the avian gut, where they're much more likely to be found in the anaerobic lumen, whereas in mammals, they're more often associated with the oxygenated mucosa, because in those species, they're far more invasive. this is roughly how the data looks in practice. We have a vast spreadsheet of the insertion sites for nearly 8,000 mutants, and we can see to the nucleotide where those insertions are located and what effect they have. This is colored in different shades here. Blue indicates a strong negative selection. Red would be strong enrichment. Yellow or orange, really not one or the other. And clearly there's a cluster of genes here that play an important role in colonization across all three hosts. But if you come out into an intergenic region, the effect is lost. So we tile through the Salmonella genome at high density, map the important regions, and can see effects, in this case, that are conserved. But in this case, that are host-specific. This is group 1 hydrogenase, one of those pathways involved in anaerobic metabolism, seems to play a more important role in the chicken than it does in other hosts. Now, this sort of analysis has been really powerful because even though it's now 2 decades since Salmonella was first sequenced, around 40% or so of Salmonella genes are still recorded as hypothetical or unknown function. And what an approach like this can do is at least tell us what role do those genes play in natural animal hosts. And this is just an example of part of the Salmonella genome where there were mutations, which are the arrows on the black lines. And these are negatively selected again by the blue color here, in all three hosts when they occur in these genes denoted in blue here. So you can see positions where mutations have no effect and then mutations which have a strong effect. And this was a cluster of genes about which we previously knew nothing, but clearly it plays an important role in farm animals, interestingly, not the laboratory mouse. So it really tells us how powerful it can be to do this sort of functional annotation in the relevant animal hosts. Now, I've mentioned some of the ways in which we can exploit this data, whether it's devising subunit vaccines or inhibitors, taking live attenuated vaccines. I think, to be fair, the problem is, where do we start? There are hundreds of genes that play important roles in cows, pigs, and chickens, and deciding which of those is going to offer the best route to control is pretty challenging, particularly for live attenuated vaccines, where we have to balance protection against persistence and pathology. And getting that magic balance of attributes right is pretty challenging. So these very same principles that I've mentioned of screening pools of mutants for phenotypes, in my group we've now begun to apply to screen pools of wild-type strains. I mentioned that we have blueprints now for hundreds of thousands of Salmonella, but what we don't have are phenotypes in relevant animals. And to test strains one by one, it's just not possible. So what we've begun to do is use this mass parallel sequencing approach to follow what happens to wild-type strains when we screen them in pools. Now, we can do that with Salmonella because there are some regions of the chromosome that are naturally variable. And if we train this deep sequencing on these polymorphic alleles, we can see which strains are present by virtue of diagnostic nucleotide polymorphisms in that region. And then the number of times we see that specific sequence read gives us the abundance of the strain. So in this case, we were interested in whether all of these serovars were virulent. In fact, this is an experiment we did with zoetis in cattle, but it's just to demonstrate the concept. What we can do is validate that this method reads out faithfully through sequencing what we know to be the composition of pools that we've assembled in the laboratory. So we can spike pools in the laboratory with this composition, where each of the colored boxes denotes the relative abundance of pools that we've made. And the correlation between the sequencing and the known composition is very strong. In fact, there's no statistical difference in any scenario that we test in. But when we take some of these pools and put them into animals, we see winners and losers. So this pool at the top here was what went down the neck of the animal, and then you can see there are some serovars that outgrow the other. There are some that we included here as control strains that we know to be avirulent in this species. They faithfully disappear. There were some that we knew to be more systemically virulent. They expand in the liver. So this sort of approach can give us a lot of information about the relative phenotypes of strains, but in a single animal here, instead of having to test each of these strains separately. And one might think about using this sort of approach when we're trying to devise cross-protective strategies. Instead of testing our vaccine or treatment against every different possible variant separately, maybe we can start to think about using pools of strains and showing they're all equally affected. Now, just before I move on to talk a little bit about host genetics, I just wanted to use this one slide to explain how difficult the challenge of functional annotation can be with bacteria when there is a great deal of genetic diversity in the pathogen. I've spoken so far about Salmonella typhimurium, and broadly speaking, many of them look alike. But if you try that with avian pathogenic E. coli, you get a very different picture altogether. There's a vast amount of genetic diversity. We were involved some years ago in sequencing strains from dominant serogroups and sequence types. This is just an example of an E. coli 078 strain from sequence type 23, a very dominant lineage. When we sequenced its genome, it was different from the next available APEC genome by 1,100 chromosomal genes and 4 plasmids with nearly 250 kilobases of different genetic information. And among all of those genes are very different virulence factors. And this is a problem when we're doing functional annotation because we tend to use a small number of prototype strains that are well studied around the world, and they just might not reflect the huge diversity that really exists out there in nature. So an important cautionary tale. So for the last 10 to 15 minutes, I'm going to change gears and talk a little bit about Campylobacter. And I won't go over the detail of this slide. It was very nicely covered earlier by Lisa and others. Suffice to say, it's a problem that we get as humans through handling or consuming contaminated poultry meat, and it's not under control. There are a number of candidate therapies and treatments that have been suggested, and some of these are partially effective, but maybe they can only reduce the burden of Campylobacter in the gut by 1 to 2 orders of magnitude in many cases. Fitted together as a combination of approaches, who knows, they might solve the problem, but for the moment we don't have effective licensed vaccines, and there are relatively few proven reliable strategies that are currently being adopted worldwide. So can you do this TRADIS methodology with Campylobacter? I've mentioned how powerful it can be for Salmonella. Well, actually, when we tried it, it was very much more difficult to get to work with Campylobacter jejuni in the chicken. And again, working with the University of Cambridge over a decade ago, we actually showed that if you take 2 wild-type strains of Campylobacter that are identical to each other, except for the presence of a short DNA barcode, actually, even those 2 wild-type strains don't compete with each other in an entirely predictable way. The population dynamic is unstable and it's unpredictable. Sometimes you could put them in different ratios and it would be the minority member that wins out of the 2. Now, this is really difficult if you want to screen mutants, because it suggests that there are processes at play that govern colonization that aren't related to whether or not you have a mutation in a particular gene. I should say that Stefan de Vries, who spoke earlier, has some great work suggesting that you can partially solve this problem by screening pools of mutants in larger numbers of chickens and then pooling the outputs from all of those birds before you analyze it. And the reference to the paper is there if you're interested. You can take a gene-by-gene approach with Campylobacter, of course. It's a much smaller genome, maybe just over 1/3 the size of Salmonella. We've done that and found roles for flagella, capsule, M-glycosylation systems, but also importantly metabolism. It's told us some things about what Campylobacter needs to grow in the chicken intestines, and one might imagine that that could be useful later in trying to find manipulations of the metabolome and microbiome. A long way off, but a starting point for analysis. What about vaccination? Well, in my laboratory, we've, over the years, reported statistically significant protection with either subunit or vectored vaccines for control of Campylobacter. So these might have subunits of C. jejuni made in a recombinant form and injected into the birds, or will express those factors in live attenuated Salmonella. We do see protective effects, but they're rather modest. They're often only 1 to 2 orders of magnitude, and they're often too late in the life of a modern broiler bird to be useful. Often we see them 7 or 8 weeks after primary vaccination on the day of hatch. It's too late. What we, and I think other laboratories, find is a lack of consistency, both between replicates of a given trial that we may conduct, but also a lack of comparison between different laboratories testing a similar strategy. This is partly because there isn't a single accepted regimen for a trial, so people often use slightly different doses, lines, and so on. But I think there is an underlying problem with inherent variation in the vaccine-induced response and how Campylobacter may or may not be affected by that. Even where we have seen protection, we know very little about the immunological basis of the protective effects. That's partly because of a paucity of tools to actually understand the functional immunology, whether by ablation or adoptive transfer or other approaches. We do have a project looking at glycoconjugate vaccines of various types, but it's perhaps too early to talk about that. So the final strategy that I'm going to talk about relates to whether or not we can harness any heritable variation that exists in birds to Campylobacter. And at the Institute for Animal Health and now at the Roslin Institute, we have housed unique inbred lines of bird that exhibit heritable differences in resistance, not just to Campylobacter, but to other bacterial, viral, and protozoan pathogens. And these are an incredibly useful resource to understand the genetic and immunological basis of protection. One of the things that we can do where there is heritable resistance or susceptibility is we can cross those two lines together, and then we can look at the genetic makeup of the progeny of those crosses and whether or not that affects their resistance to colonization. And we've been doing that over the years for Campylobacter jejuni. Using these two lines, 6 and N, and as you can see from this study, they exhibit quite a profound difference in colonization by C. jejuni. It's often around 2 to 3 orders of magnitude. But what we observe is that this is independent of bacterial strain. We've reliably seen it both with our populations and populations held elsewhere. So you might think, oh, problem solved, Mark. Why can't we just eat these birds? Well, of course, they're inbred. They're a layer line, and they'd be absolutely useless in any sort of production context. But as a genetic resource for understanding the genomic architecture of the resistance trait, then they're incredibly powerful. Previously at the Institute for Animal Health, we had studied a back-cross population, and working with collaborators at INRA, we performed studies in an advanced intercross population of these two lines. I'm not going to go too much into what we found. The data are published and the references are at the bottom. But we've measured the Campylobacter burdens in the ceca of these animals, and then we've sampled genetic variation at tens of thousands of positions along the chicken genome. We're looking here for single nucleotide polymorphisms at specific positions. And if you plot out all of these nucleotide variants and then look at their statistical association with the measured trait, in our case, Campylobacter burden in the ceca, then you can see on these so-called Manhattan plots that there are some positions in the Salmonella genome where you go above a magic threshold of statistical association between variation at these positions and resistance to Campylobacter. These are so-called quantitative trait loci, or QTL, and in these populations, we found 4 genome-wide significant QTL for Campylobacter resistance. Now, we've been fortunate to partner with Aviagen to take this the next step forward and ask, is there a heritable component to resistance to Campylobacter in commercial broiler populations? We've been studying inbred lines and it's been useful, but how relevant is it to a modern meat bird? Working over a number of years with great support from Aviagen, we've performed a genome-wide association study with over 3,000 commercial broilers. We've got high-quality phenotypes, again, in terms of cecal CFU of Campylobacter, and high-density SNP genotypes for about 90% of the birds that we looked at. So 2,700 birds in this population analysis. We've accounted in the GWAS model for sex and seasonal sampling effects. And again, we've identified genome-wide significant QTL. What was really cheering is that some of these are the same as those that we found in the progeny of crosses from the inbred lines. So that tells us something about how relevant those lines still remain to mapping resistance determinants. We've since gone on using Scottish Government funding to perform some RNA sequencing of cecal mucosa of birds at the extremes of colonization phenotype. These were selected to have the resistant or susceptible genotypes and correspondingly to have low or high Campylobacter burdens. And remarkably, when we do this, we find very few differentially transcribed genes in the ceca of these resistant or susceptible birds. If you apply a very strict cutoff, it's only 4 or 5, and remarkably, 3 of them fall within the QTL intervals. So it may be that there's something within those regions of the genome, it may be at the expression level rather than the sequence level, that underlies this differential resistance. And we're working at the moment with Aviagen to further dissect that. So there is a heritable component to Campylobacter resistance, but is it going to be useful? Well, the estimates of heritability are relatively modest. Maybe only 11% of the trait is explained by bird genetics, and the rest is everything else. That's a relatively low component to selectively breed on, but it nevertheless is possible to do that, we predict, without compromising other breeding goals, including feed conversion, body weight, and gut health indices. Unfortunately, the resistant-associated variation that we looked at was already quite prevalent in the commercial populations that we studied, but it may be that these information will be useful in guiding selective breeding decisions in other populations and lines. So just going to touch very briefly on whether or not there might be a component for the microbiota here. Clearly, host resistance is partly determined by the bird line, but how much of that is bird genetics and how much of that might be distinct microbiota? I won't go too much into how these studies were done. We've heard a lot about microbiome analysis today, so you'll be familiar with the methodology. What we've done here is try some flora transplants between line 6 and N. And we've done this using flora from donor birds that were 3 weeks old, the age at which Campylobacter resistance was previously reported. And we've given that donor flora either in a homologous way, line 6 into line 6, line N into line N, or a heterologous way by swapping them over. And then after 2 attempts at doing the flora transplant, we've challenged at day 21 with Campylobacter jejuni. And what you may just be able to see here is that the resistant line stays resistant, whether it had flora from line 6 or line N, and the susceptible line stays susceptible no matter where the flora came from. And that might not be surprising when you look at the microflora from the donor birds, where although there was a slight but non-significant difference in the community structure when we analyzed the 16S amplicon types by principal component analysis, it's not significant. And if you look at the phylum and family level, we really can't see anything striking about the microbiota in these 2 lines of birds that are quite evidently different in resistance. You can track these things over time as well. One of the most striking things here is actually just how dominant E. coli is. Despite being a minor constituent of the donor microbiota, when you look shortly after colonization, this is just a day, E. coli by far the most dominant of the proteobacteria there, and then there rapidly is a return to some other types. You can see some line-specific operational taxonomic units. This is something from the genus Ocelospira, but you have to look pretty hard to find these effects. This is one that appears somewhat more prevalent in the resistant line. But of course, discerning cause and effect when we look at these associations is pretty challenging. So that's all I'm going to say. I'm going to try to keep us broadly to time. I hope I can convince you that we do still need to study microbial pathogenesis, because for many avian and zoonotic pathogens, simple interventions are not fully effective, or at the moment they're ineffective. And as I mentioned, we live in a post-genomic era now where we have no shortage of information on what pathogens look like, but we have relatively little information on what the encoded genes do during infection, and also how the blueprints of these strains relate to their virulence in target hosts. And hopefully I can convince you that there are some fantastic new approaches that rely on deep sequencing to tackle that. These same genome-scale mutagenesis approaches are now becoming feasible for eukaryotic cells. In the same way as you can target every gene in Salmonella, you can do that now with CRISPR-Gecko libraries in cell lines. And that's going to be a very powerful technology for looking for host genes that may determine responses to pathogens instead of doing, or perhaps as well as doing, genome-wide association studies. The gap I see that we really need to address in studying host-pathogen interactions is addressing the gap we have with tools to do functional immunology with the bird immune system. We really could do with some edited and transgenic chicken lines that will be fantastically powerful resources for looking at the role of different components of the avian immune system in protective responses. So that's all I'm going to say. I've got very many people to thank, from both Roslyn and formerly the Institute for Animal Health, my various collaborators and funders, and my team. And I thank you for your attention. I'll be happy to take any questions.